കണ്ടായിരുന്നോ മെസ്സേജ്?
സൂര്യകുമാർ ഷെയർ ചെയ്യാനായിട്ട്
സൂര്യനാരായൺ
എന്താണ് പറ്റുന്നുണ്ടോ? ഞാൻ മഹേഷിനോട് പറയാം.
നമസ്കാരം.
നമസ്കാരം. നമസ്കാരം.
നമസ്കാരം.
സൂര്യനാരായണൻ നമസ്കാരം.
നമസ്കാരം.
ഇപ്പൊ എനിക്ക് പ്രസന്റ് ചെയ്യാം.
വെബിനാർ സ്റ്റാർട്ട് ചെയ്യാം. സൂര്യകുമാരെ പ്രാർത്ഥനയോടുകൂടി വെബിനാർ സ്റ്റാർട്ട് ചെയ്യാം.
ഓം സഹനാവവതു സഹനോ ഭുനക്തു സഹ വീര്യം കരവാവഹൈ തേജസ്വിനാവധീതമസ്തു മാ വിദ്വിഷാവഹൈ. ഓം ശാന്തി ശാന്തി ശാന്തി.
എല്ലാവർക്കും നമസ്കാരം. ഇന്ന് നമ്മുടെ വെബിനാർ നയിക്കുന്നത് മാസ്റ്റർ സൂര്യനാരായണൻ. സൂര്യനാരായണൻ ശ്രീ രാജേഷ് കുമാറിന്റെയും അർച്ചന നാരായണന്റെയും മകനാണ്. 21 വയസ്സാണ് പ്രായം. 21ാം തീയതിയാണ് വെബിനാർ അവതരിപ്പിക്കുന്നത്. ഒരു സ്ട്രൈക്കിംഗ് പോയിന്റ്. സൂര്യനാരായണൻ ഇന്ത്യൻ ഇൻസ്റ്റിറ്റ്യൂട്ട് ഓഫ് സയൻസ് എഡ്യൂക്കേഷൻ ആൻഡ് റിസർച്ച് എന്ന സ്ഥാപനത്തിൽ ബാച്ചിലേഴ്സ് ആൻഡ് മാസ്റ്റേഴ്സ് ഡിഗ്രി ഇൻ സയൻസിൽ 3ാം വർഷ വിദ്യാർത്ഥിയാണ്. ഏറ്റവും വലിയ നേട്ടം സൂര്യനാരായണ അടുത്ത കാലത്ത് ഉണ്ടായത് ഇന്റർനാഷണൽ സിന്തറ്റിക് ബയോളജി കോമ്പറ്റീഷൻ എന്ന പ്രോജക്റ്റ് ഇന്റർനാഷണൽ ജനറ്റിക്കലി എഞ്ചിനീയർഡ് മെക്കാനിസം എന്ന സ്ഥാപനം വഴി നടത്തിയപ്പോൾ 9 മാസം നീണ്ടുന്ന പ്രോഗ്രാമായി. അതിൽ ഗോൾഡ് മെഡൽ കരസ്ഥമാക്കാൻ കഴിഞ്ഞു എന്നുള്ളതാണ്. ഇത് വാസ്തവത്തിൽ വളരെ വലിയ നേട്ടം തന്നെയാണ്. അഭിമാനാത്മകമായ നേട്ടമാണ്. വളർന്നു വരുന്ന തലമുറകൾക്ക് വളരെ ആവേശം പകർന്നു നൽകാൻ കഴിയുന്ന ഒരു നേട്ടമായിട്ടാണ് ഞാൻ ഇതിനെ കാണുന്നത്. അപ്പം സൂര്യനാരായണ വളരെയധികം ഉന്നതങ്ങളിൽ എത്താൻ കഴിയട്ടെ എന്ന പ്രാർത്ഥനയാണ് എനിക്കുള്ളത്. അതോടൊപ്പം ഈ വെബിനാർ എന്ന് പറയുന്നത് സൂര്യനാരായണനെ സംബന്ധിച്ച് നല്ലൊരു എക്സ്പോഷർ കിട്ടുന്ന കാര്യമാവുകയും ഒരു മോട്ടിവേഷൻ ആവുകയും ചെയ്യട്ടെ എന്നുള്ളതാണ് എൻ്റെ പ്രാർത്ഥന. ഇത് കൂടുതലധികം ഇതിലോട്ട് പോകുന്നില്ല. ഈ വെബിനാർ പ്രസന്റ് ചെയ്യുക, അതിനുശേഷം സംശയങ്ങൾ ആർക്കേണ്ടതുണ്ടെങ്കിൽ അത് ചോദിച്ചിട്ട് അതിനുള്ള പരിഹാരവും സൂര്യനാരായണ നിർദ്ദേശിക്കുന്നതായിരിക്കും. അപ്പൊ ഇത് വളരെ ലൈവ്ലി ആയിട്ട് വരട്ടെ. കാരണം ഇന്ററാക്ഷൻ എന്നുള്ള സംഗതിയിലുണ്ട്. വന്നിട്ട് വളരെ ഇന്ററാക്റ്റീവ് സെക്ഷൻ ആയിട്ട് വരണം എന്നാണ് നമ്മളൊരു കൺസെപ്റ്റ്. നമ്മുടെ വെബിനാറുകളിൽ ഏറ്റവും പ്രായം കുറഞ്ഞ വ്യക്തി സൂര്യനാരായണൻ. ഇതുവരെ നമ്മൾ പങ്കെടുത്ത എല്ലാ വെബിനാറും നോക്കിയാൽ ഏറ്റവും the youngest man among the persons who conducted the seminar webinar is Surya Narayan. And I'm proud of you, Surya Narayan, and I'm sure that you will be able to present a very inspiring talk to the listeners. And let us be motivated together. You motivate us, and we motivate you. So when that motivation goes together. So I request Surya Narayan to present the webinar, and everybody else, those who are not speaking, they should mute the mic. And so this program is totally recorded, and it will be in the YouTube also. So Surya Narayan, you please start your presentation.
Can you see my screen?
Yeah, we can.
You can only see the slides, not my notes, right?
Only the slides.
Okay. Hello, my name is Surya Narayan. I'm a third year BSMS student at the Indian Institute of Science, Education and Research, Pune. I'm currently working on my physics major. I, along with a few of my classmates, participated in an international synthetic biology competition. My team won the gold medal in the competition. Today, I'll be talking about our idea, which won us the gold medal, and about synthetic biology as a whole, what it is, why people do it, what all you see around you are synthetic biology, and how you can get involved with synthetic biology. It might seem odd that a physics major is talking about synthetic biology, but it is one of the most amazing fields out there where you can see the results of your work almost immediately. What is synthetic biology? The textbook definition goes like this. Synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. We are going to break this sentence down and understand all the ideas it is hinting at. But before we do so, there are a few basic facts that everyone needs to know. Anything that has life, and in some cases doesn't, are called organisms. Organisms can be made of either single cell, as in the case of bacteria, or multiple cells, as in the case of plants and animals. Everyone here must be familiar with the with people saying, "You look like your dad. You look like your mom. You have your mom's eyes. You draw well, just like your mom." And it is true. Humans do inherit their parents' looks and to some extent skills. But how can we explain this from a biological perspective? There has to be something that carries the information about how one looks and all the talents the parent possesses. Biologists, and nowadays common folks as well, like to call this something genes. Genes carry all sorts of hereditary information. Genes are responsible for us looking like our parents. Genes are made up of chemical compounds called deoxyribonucleic acid, or in short, DNA. You might wonder how exactly can a non-living compound such as DNA be capable of storing hereditary information? Sequences. DNA is further made up of four compounds called bases. There are four different kinds of bases: adenine, guanine, cytosine, and thymine. When you create a sequence using these four bases, a certain amount of information is stored in it. The sequence can be hundreds, thousands, or even millions of bases long. How information is stored in DNA is very similar to how it is stored in a computer as ones and zeros. When a child is born, they inherit DNA from both their parents, and this is the reason why they look and behave like their parents. Same goes for all organisms, be it bacteria or plants. I said earlier that the reason you look like your parent is because of the information stored in genes. Well, it is true, but it doesn't capture the importance of genes all that well. Genes are like an instruction manual for your body. Genes dictate what color your eyes will be, how you will digest food, how tall you'll grow, and so on. You might have heard your grandparents talk about how they're afraid they might get diabetes because their parents had it. Well, this can be explained using genes. Genes store information on how well your insulin production is going to be. And since insulin is what helps use up all the sugar that you eat, insulin production capabilities play a major role in whether you're prone to or not get diabetes at some point in your life. But this does not mean that your parents or grandparents are diabetic, implies that you will also be diabetic at one point. What you eat does matter. Now that we've gotten an idea of what an organism is, how information is stored in an organism, and how it is passed on from parents to offsprings, let's look at how one can manipulate this information and the passage of information, and if we can create our own DNA sequences from scratch. This is where synthetic biology comes in. Let's revisit the textbook definition of synthetic biology. Synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. Redesigning and engineering are the keywords that bring about a world of difference between biology and synthetic biology. Just like how we can edit data stored in our computers, write our own data, and delete data from computers, one can edit, create, and delete DNA sequences to bring about changes in the appearance and capabilities of an organism. Humans are made up of millions of cells, and not all of them are the same. Your skin cell is different from your nerve cell, which is different from your liver cell, which is different from your hair cells, and so on. All cells in your body contain the exact same copy of DNA. Imagine if you were to edit, add, or delete just one of the cells, nothing would happen exactly. Nothing would happen because there are many other copies of DNA in every other cell. You'll have to do the same changes that you did to the one cell to all the cells, and the procedure to do so will be different because of the varied nature of cells. This is the prime reason why scientists restrict themselves to working with single-celled organisms like bacteria. Although there are experiments going on with mice, but that is something I'm not going to be talking about right now. You might be wondering why I haven't mentioned the exact ways of performing these changes to DNA, which we are going to be calling as genetic modifications from now on. This is because the science behind it might get a bit too technical and might just be a bit too boring. If you are interested to know more about it, you can visit a cute video series that my team and I curated and made throughout the course of our project to help beginners in the field of synthetic biology. Now, I will be taking up a few examples on the applications of synthetic biology, and I will try to explain the science behind each of them, although very abstractly. First one is bioisoprene. Till a few years back, we had been using natural rubber, which was obtained from rubber trees. You might already see the limitations of this. Trees are limited and they require a lot of care and a lot of land. Recently, we've switched to synthetic rubber, which is modified version of natural rubber. It is known to be stronger and more sturdy than natural rubber. Isoprene, also known as the natural rubber, is used in the production of synthetic rubber, which is used in tires, sports goods, and erasers, etc. Currently, synthetic rubber is obtained by burning fossil fuels. In this age of increasing carbon dioxide content in the atmosphere, this is frowned upon. This brings about a necessity for other, more cleaner ways of producing this commodity. DuPont, together with a Goodyear tire and rubber company, is currently working on the development of a reliable, high-efficiency, fermentation-based process for the bioisoprene monomer. And synthetic biology has played an important role in making this undertaking a reality. What they've done is to traverse all the genes of the rubber tree and identify the genes responsible for the production of the so-called natural rubber, create the sequence from scratch in the lab, and put it into a microorganism like bacteria. This bacterium is then capable of producing bio-based rubber, which can then be modified again to produce synthetic rubber in the same bacterium. This bacterium is then capable of producing bio-based synthetic rubber, which would go a long way in reducing carbon dioxide content and reducing the harm to trees. Second one is acrylic. Acrylic is an important petrochemical used in the wide range of industrial and consumer products. Acrylic ingredients make paints more durable and odor-free, adhesives stronger and longer-lasting, diapers more absorbent and leak-proof, and detergents better able to clean clothes. Today, petroleum-based acrylic is an $8 billion global market. OPX Biotechnologies is developing a renewable bio-based acrylic to match petroacrylic performance and cost, but with a 75% reduction in greenhouse gas emissions. Here again, we take genes from multiple organisms and put it into one so that it meets the required conditions to surpass the existing petrochemical synthesis methods. The third one I'd like to address is surfactants. Surfactants are one of the most useful and widely sold classes of chemicals because they enable the stable blending of chemicals that do not usually remain associated, like oil and water. A prime example of this would be all cosmetics like moisturizers, primers, foundations, etc. Today, nearly all surfactants are manufactured from either petrochemicals or seed oils, such as palm or coconut oil. Worldwide production of surfactants from petrochemical annually emits atmospheric carbon dioxide equivalent to combustion of 3.6 billion gallons of gasoline. Production from seed oil is greener, but this is a limit to the amount of seed oil that can be produced while protecting the rainforest. To address this problem, Modular has developed microorganisms that convert agricultural waste material into useful new surfactants. Modular has developed an engineered has developed and engineered microorganisms that converts soybean hulls into surfactants for the use in personal care products and other formulations. The hull is a woody case that protects the soybeans, and it cannot be digested by humans or other animals such as pigs. The US produces about 70 billion pounds of indigestible soy carbohydrates annually, and Modular seeks to upgrade this underutilized material by converting it into a variety of useful new chemical products. Synthetic biology is in medicine is another interesting topic I'd like to bring up at this point. Till now, you've seen how various genetic modifications made to microorganisms can bring about changes in them that could prove useful to us. When it comes to synthetic biology in medicine, it's more about detecting, warning, and preventing rather than manufacturing. One key aspect of synthetic biology that I forgot to mention before was that in order to bring about modifications in genes and gene sets of organisms, one has to know the genes and gene sets almost thoroughly. Traversing and recording the entire gene sets of organisms is called genome sequencing, which is a tool of synthetic biology. Going back to synthetic biology in medicine, one can engineer bacteria to sense pathogens present in patients' saliva, urine, or blood tests. These can be used as various medicinal tests instead of the tests that we currently conduct. One can also engineer microbes to detect the infection in the body and deliver drugs to the corresponding location. This way, surgeries can be limited to just simply injecting the so-called microorganism into the bloodstream, and it can deliver the drugs to the targeted location pretty safely. This is essential. This is an essential. One can also find potential targets in the pathogen's gene sets that could kill it if tampered with. This is an essential idea behind creating drugs. You create drugs that target the particular set of genes that, if tampered with, the pathogen dies. Since I'm not very familiar with the workings of synthetic biology in medicine, I'm not going to talk much about it. Everyone knows this is another thing that I should probably address in the topic of synthetic biology. Everyone knows about corona pandemic. Everyone knows that it was an outbreak from some lab that was working on viruses. So what? Will everything that is being worked on have a potential to turn into a pandemic? The short answer is depends on what you're working with. There are two kinds of microbes, one that can survive only in labs and ones that survive outside. Most of the times, these lab organisms die when exposed to their outside environment. But that is not the case always. And people also work on organisms that exist in the environment, not just the ones that exist in the lab. So there has to be some protocol or a rule book that prevents pandemics from happening every other day. This is where biosafety comes in. Biosafety is defined as the discipline addressing the safe handling and containment of infectious microorganisms and hazardous biological materials. The practice of safe handling of pathogenic microorganisms and their toxins in the biological laboratory is accomplished through the application of containment principles and the risk assessment. In layman's terms, biosafety is a rule book that prevents the bacteria from infecting the researcher or anyone else. It guides researchers on how to use the lab facilities, how to make sure everything is sanitized before and after working on the specimen. Many times when working with microbes, you have to work with carcinogenic reagents. Carcinogenic reagents are reagents that can potentially cause cancer to whoever it may come in contact with. Safety then becomes of utmost importance. What if the person working with the carcinogenic substance touches it by hand and then goes about shaking hands with people, touching the rails of stairs, touching plates in the kitchen? Everyone who will then interact with the rails, the plates, or with him will be exposed to carcinogenic substances and are at a risk of potential cancer. Take another example where you're working with an infectious bacterial disease. You did all your experiments with bare hands instead of doing it with disposable gloves like a normal person. Now you're at the risk of getting the infection, and so is everyone around you. To avoid these kinds of mistakes or pandemics, we follow the safety protocols by laid down in the rule book called Biosafety. The picture shown in the slide over here is what is called a hood. This is where all the biological experiments are done. This is everything that is taken into the hood and outside is sterilized before and after the action. Any experiment that is done inside the hood. People sterilize themselves before and after working in the hood. Uh, I'll list out a few of the biosafety protocols that my team followed throughout the project, and it hopefully gives you some idea of how the project is and how biosafety can prevent pandemics like this in the future. Laminar flow hoods, which is what is shown in the picture, were put under UV radiations for 20 minutes multiple times a day. This is to kill any microbe that might have entered from outside or to kill any microbe that might have escaped the vessel they were meant to be stored in. The hood was also sterilized with 70% ethanol solution. This is a second layer of security to kill unwanted microbes in unwanted places. Everyone knows how alcohol can be used to cleanse wounds. The similar principle is applied here. All reagent vessels introduced into the hood, for example, small tubes of antibiotics, were placed in one corner in the front of the hood, and samples were taken from there via sterilized micropipette. This, in a way, reduces the spread of all the bacteria that could potentially end up being harmful, or in other cases, could be unharmful, could be of no harm as well. Before any sterile liquid/culture was transferred from one vessel to another, the mouths of the vessel were sterilized using a flame from a burner in the flow hood. This, in a way, protects um, alien bacteria from mutating and then potentially becoming harmful in the future. No food or drink was allowed in the proximity to the sterile spaces in the lab. I hope this gives you a good enough idea on the safety measures that is taken around the world to make sure pandemics don't happen every other day. Now we are going to talk about the competition and the organization that hosts the competition that my team and I participated in. The International Genetically Engineered Machine, or Foundation, or IGEM for short, is an independent, non-profit organization dedicated to the advancement of synthetic biology. IGEM's biggest program is the IGEM competition, which my team and I participated in last year. The IGEM competition gives students the opportunity to push boundaries of synthetic biology by tackling everyday issues facing the world, made up of primarily facing the world. Made up of primarily university students, multidisciplinary teams work together to design, build, test, and measure a system of their own design using interchangeable biological parts and standard molecular biology techniques. Every year, nearly 6,000 people dedicate their summer to iGEM and then come together in the fall to present their work and compete at the annual Jamboree. But before we go further into the topic of competition, I would like to address a database hosted by the iGEM Foundation called the iGEM Registry. It is a repository of all the creations and blueprints produced by all the teams throughout the years. It is open to the public. Anyone can add a new part, anyone can order parts from it. The only thing is one has to follow a certain set of rules while preparing a part that needs to be added into the registry. Enforcing such rules has made it very easy for people to just take the parts from the registry and work with it, because you can follow the same rule book in order to modify the part into something that you want. All of this is very technical when if you want an in-depth explanation, and therefore I will not be diving into the in-depth explanation. All of this is explicitly mentioned in our YouTube series if you want to check it out. Going back to the competition, iGEM began in January 2003 as an independent study course at MIT, where students developed biological devices to make cells blink. The course became a summer competition with 5 teams in 2004, then continued to grow to 13 teams in 2005, and now has expanded to a whooping 353 teams in 2019, reaching over 40 countries. The competition was originally aimed at undergraduate college students and now has grown to include graduate and high school students. By 2011, the growth of iGEM around the world sparked the first regional jamborees, with the highest scoring teams from each region proceeding to the World Championships in Boston. By 2014, iGEM outgrew their home at MIT, and in an effort to reunite the whole iGEM community at a single event once more, the Giant Jamboree was held at the Hyannis Convention Center in Boston. This year it was meant to happen in Paris, but due to Corona and other reasons, we couldn't attend the giant jamboree, and therefore it was online. We had regional jamborees, and the jamboree in India was conducted in Goa. Now I'll be talking about my experience and my team's experience throughout IGEM. My team started working on the project in March of 2021. Our college produces teams participating in IGEM almost every year. You apply for it like any job post and you go through assignments, interviews until you make it to the team. Usually the team comprises of second and third years, and our team comprises of the same. We were a team of 12 people, 10 second year students, one third year, and one integrated PhD student. Some of us did not even have a background in biology, let alone synthetic biology. So naturally, a lot of our time was spent learning new techniques and topics. I myself had zero background in biology or synthetic biology, and I ended up becoming the modeling head of this team. Apart from the science part, we also had to get accustomed to working in a team setting. Most of us were friends from before the competition, so it was easier for us to get along. The initial months were just studying about the synthetic biology, the techniques involved, and the uses of it, and trying to come up with an idea that satisfied the criteria put forward by the competition hosts. The hosts award medals depending on the amount of work and quality of work. They clearly mention a list of tasks a team has to accomplish in order for them to get a gold medal. Without careful planning, you might just fall short of a few handfuls of items, and that could cost you the gold medal. So our initial months were all also included us figuring out our game plan. We ended up choosing an idea that deals with global warming and lack of green ways to produce fuels. Before we go into the details of our idea, it'd be better if I gave you a reason why we chose our idea, and this could potentially be the part that would inspire people to take up fields like synthetic biology because your lives are at stake. Carbon dioxide concentration levels in the atmosphere are higher now than at any other time in the human history, and they are only projected to rise due to human activities. Carbon dioxide is a greenhouse gas which plays a crucial role in regulating the surface temperature of the planet. As CO2 accumulates in the atmosphere, it leads to a net decrease in the average global surface temperatures, a net increase in in the average global surface temperatures. My bad. We are already beginning to experience the effects of climate change due to global warming. From more extreme rainfall, droughts, cyclones, and wildfires to extinction of numerous plant and animal species. The globe has already warmed by over a degree centigrade above pre-industrial levels. In order to limit global warming to 1.5 degrees centigrade above pre-industrial levels, greenhouse gas emissions must fall by 45% by 2030, and we must reach net zero emissions by 2050. A carbon feedstock is a source of carbon that can be used as a raw material for manufacturing. Most carbon-based industrial chemicals rely on fossil fuels like coal, crude, and other raw materials for their manufacture. Like I mentioned before, in the case of isoprene, in the case of synthetic rubber, acrylic, and surfactants, a lot of carbon-based industrial compounds release a lot of greenhouse gases when um, during the manufacturing process. However, the use of coal and oil for the production of these chemicals emit large amounts of carbon dioxide and other greenhouse gases that warm the atmosphere and the oceans. The chemical and petrochemical industry is the third largest source of industrial CO2 emissions, generating a whopping 1.5 gigatons of CO2 annually and accounting for 18% of the direct industrial CO2 emissions. Now would be a good time for you to recollect all the synthetic biology that you came across in the past few minutes. You'd only need a very basic idea. You will not need a very in-depth idea. And a sustainable alternative to fossil fuel-based carbon feedstock is to make use of biomass as a carbon feedstock. Heterotrophic bacteria can be engineered to consume carbohydrates from biomass and convert them into industrially important metabolites. However, relying on plant-based biomass like corn and sugarcane as raw materials for industrial scale chemical synthesis could compete with food production and advance environmental degradation. Plant cultivation is often resource intensive, requiring a lot of land, water, and fertilizers. A truly sustainable carbon source would need to be far more efficient. Cyanobacteria are photosynthetic prokaryotes. Prokaryotes are a type of single-celled organisms that are far more efficient than plants at using sunlight to fix atmospheric CO2 into biomass. They can convert as much as 9% of the solar energy received into biomass compared with only 0.5 to 3% for plants. They also require a fraction of the space and resources needed to cultivate plants. As you know, bacteria are very small, and a handful of bacteria can produce the same effect as maybe a whole tree, and therefore the space they require would be very less, like exponentially lesser than what plants would require. Cyanobacteria are more amenable to genetic manipulation than eukaryotic algae or plants and can be grown conveniently in a photobioreactor on unproductive land, wastewater, and flue gas. As a solution, my team and I designed a sustainable biomanufacturing method to synthesize biobutanol from light and carbon dioxide using a co-culture of two bacteria. Cyanococcus elongatus UTEX2973. For simplicity, we'll be calling the cyanobacteria, and E. coli. Our solution, and E. coli is a type of bacteria. It's the biologist's favorite bacteria to work on because it is very easy to genetically manipulate. Our solution would minimize carbon emissions by not only capturing existing carbon emissions present in the atmosphere, but will also prevent new emissions from being generated at the source by reducing the current reliance on carbon emitting petrochemical feedstock. As long as it is UTEX2973 is a fast-growing variant of cyanobacteria, which accumulates sucrose intracellularly, which means it accumulates sucrose within the cell on exposure to salt stress. This sucrose is obtained from the atmospheric carbon dioxide fixed by the bacterium during photosynthesis. We engineered it to continually secrete the sucrose it generates. As elongated UTEX2973 can secrete nearly 90% of its absorbed carbon as sucrose, as opposed to just 15% in sugarcane. This secreted sucrose can function as a renewable carbon feedstock for chemical synthesis via E. coli. E. coli can be made to consume the sucrose generated by cyanobacteria as its sole carbon source. With an appropriate set of modifications, E. coli can be made to produce a desired metabolic product, in our case, butanol. In short, our idea came out came about because of the rising concerns of global warming, increased CO2 concentrations in the air. We engineered cyanobacteria to produce sucrose from the carbon dioxide present in the air and engineered E. coli to produce butanol from the sucrose produced by the cyanobacteria. This is a very simplistic pictorial representation of our idea. The green thing is the cyanobacteria and the orangish yellow thing is the E. coli. That's the theory behind our idea. How exactly we bring it into the real world is a whole different story, which I'm not going to be telling in much detail because it is very technical. We'll be bringing this into the real world in the form of a bioreactor. A bioreactor is an apparatus in which a biological reaction or process is carried out, especially on an industrial scale. Bioreactors are normally uh, very well sealed so that the bacterium does not escape into the environment causing biohazards, and this in a way accounts for the biosafety part of our project in a real life implementation. You might be thinking that's cool and all that you made all this happen, but what exactly is the use of this to a common man? Well, the answer to that would lie in the uses of butanol or biobutanol in our case. The primary use of biobutanol is a fuel in internal combustion engines. Although ethanol and biodiesel are preferred biofuels, it is expected that biobutanol will gain prominence in the next 5 to 10 years. This is owing to the biobutanol's higher energy content, low volatility and corrosiveness, and its higher compatibility with existing vehicles and oil industries. You can basically use it as a drop-in replacement for petroleum-based butanol. Increasing urbanization globally has led to a rise in the demand for butanol-based products for the purpose of infrastructure and constructions. It is also heavily used for the manufacturing of coatings for several end-user industries and is an essential intermediate in the production of butyl acrylate and adhesives. Its ability to use it as an additive has also increased demand in the pharmaceutical industry. So in essence, this plays a major role either directly, as in the case of fuels for engines, or indirectly, as in the case of adhesives and additives. That's no science for the day. Even my brain is too saturated with science. So let's move on to more lighter and more humane aspects of the project that we did. Um, First one being human practices. iGEM makes a big deal out of human practices. Human practices comprises of anything that we, the team, do to get common folk aware of our idea and synthetic biology as a whole. This talk I'm giving right now would be considered as part of our human practices if we were still in the iGEM window. Throughout our project, we made efforts to get through to the common people, alert them about the alarming state of our environment, and teach them about synthetic biology and our idea. Our initiatives primarily focused on students from children as young as 5 years old all the way to college students. We did this for two main reasons. First, being that the climate change is going to be a significant concern in the future, one that the youth of today will inherit, making it paramount to raise awareness for they will be the leaders of tomorrow. The second being that the COVID-19 pandemic made education digital. We reasoned that it would be easier now, more than ever, to reach a larger audience, irrespective of geography, language, or background, since more students were engaging in educational resources online than ever before. We learned that only 26% of the school children in India are instructed in English, and thus translated a lot of our outreach material, which was originally in English, to regional languages. We also organized an audio webinar for visually impaired students and made a silent film for deaf or hard of hearing students to further ensure that the spoken language was not a barrier. For very young students, we designed an interactive and fun activity book about climate change. The picture you saw on the previous slide was the event where we sat down with kids, with underprivileged kids from around our college and helped them through the activity book, making them understand the conditions that the future will have to face and making sure that they know what is in store for them and what changes they can possibly do to avoid such a grim future. Um, for high school students, we designed a survey on climate change and synthetic biology to better understand their perceptions, since they would have likely come across these issues before. We use the results of the survey to guide us in preparing targeted outreach materials such as educational video series titles in Bio 101. This was the video series I was referring to when I said it helps as a starter pack for beginners in synthetic biology. We also hosted an art and essay competition for students of all ages to promote discussions and critical thinking of the topic of climate change. To reach out to students from marginalized communities, we collaborated with Disha, which is a student run social outreach initiative at our institute that aims for socioeconomic equity through education to organize an educational webinar. We also engage with the general public through articles in newspapers and televisions and infographics and posters on our social media. This is a sort of collage of all the outreach activities that we did throughout our iGEM period. The top right being the YouTube series uh, that acts as a starter pack for anyone who wants to get involved in the technicals of synthetic biology. The bottom right being the art and essay competition for students so that they think more about climate change and how we can represent it, how we can tackle it. The top left are newspaper clippings of all the articles that mentioning us and our project. And the bottom left is an audio webinar that was conducted for visually impaired students. Um, Visually impaired students. Once we got a word out through to the common people, we needed to get ourselves some strong footing, credibility, and financial foundation. This is where the next aspect of our project comes in, integrated human practices. This mainly consists of us talking to the target consumer market of our product and understanding what exactly they require. Since our end product is a bioreactor that captures carbon from emissions and produces a biofuel, our stakeholders would primarily be companies in the industry segment. During the IGEM window, we couldn't get a lot of contacts, and we couldn't and therefore we couldn't exactly get a hold of stakeholders. But what we managed to do in place was to put in enough effort and research to come up with a solid plan that could potentially get the stakeholders involved with us. Getting stakeholders involved with us is very important because that would imply that we are contributing in our own ways to reducing the carbon content in the atmosphere, and that was the sole purpose with which we started such a project. Our plan revolves around carbon credit. A carbon credit is a permit that allows companies that holds it to emit a certain amount of carbon dioxide or other greenhouse gases. One credit permits emissions of a mass equal to 1 ton of carbon dioxide. If a company has excess credits, they can trade their credits with other companies for other benefits. There is a catch to all this, though. The number of credits provided to the company decreases as time passes. Since the ultimate goal of introducing the carbon credit system was to prohibit companies from unreasonably pumping out carbon dioxide into the atmosphere, such a catch would make sense. This is where our project comes in as a takes a spotlight for companies. Now imagine that the trades a company would be able to make if they had access to our project. In an ideal scenario, they can keep reusing the carbon emitted from the industry to make more fuel, which our project makes. It makes biofuel. It makes biobutanol. Burn the fuel for more energy. Use the energy to get whatever work they want to get done. Capture the carbon from the burning of the biofuel and recycle it to produce more biobutanol. It's basically a perfect circle in an ideal world, and companies would have enough credits to get all the benefits in the world they desire. Obviously, this is very idealistic, but I said this just to give you a good sense of the potential of our project. But what is the point of me saying all of this? The competition is over. It's not like we're going to carry on with the project. Not quite. IGEM offers their teams a chance to take their project forward as a startup. In fact, they encourage teams to take their projects forward. They even have programs in place that provide funding to teams that put forward amazing pitches. Naturally, seeing all the hype, some members of my team also wanted to take our project forward as a startup. As of now, we plan on finishing all the pending ideas and models that we had during our IGEM window, some of them being creating an actual bioreactor, optimizing for maximum efficiency, calculating the total effect of the carbon concentration in the atmosphere due to our impact. Once we are done working on those ideas, we plan to prepare a pitch for the IGEM program that could potentially get us some funding. We are also trying to find other sources of funding through stakeholders, corporations, and personal contacts. Within the next couple of years, we plan to bring the project to a technology readiness level of 5, which would mean that corporations could potentially use it very soon. With more and more companies being forced to work to save the environment and fund science projects, our project presents itself as a fruitful opportunities for companies. Companies can fund their project and call it CSR. Companies can fund our project and call it CSR funds, and can also expect a product within a few years that potentially give them an insane advantage in the carbon trading sector. Um, Enough about my project and about the impacts of my project. Let's talk about how students in school and in maybe even their first years of their degree course can get into the field of synthetic biology and can do things like me and my team and I did. So let's talk about how students can get involved in synthetic biology. As I said before, I'm a student studying in Iisc Pune, and I'm doing my third year. Students across India are generally told you should aim for IIT. IIT is the topmost college, and I'm not refuting that. This is great. IIT is an excellent institute, but engineering isn't the only job out there. Since students are getting more and more freedom, we need to give them all the options available to them. And engineering isn't the only option. IITs are great, but it's just engineering. Like there are other jobs that could potentially have more impact in the world. The world needs researchers just as much as engineers. Now that the engineering sector is so populated, unemployment rates have skyrocketed. Researchers, on the other hand, are so scarce that even if you have a small degree, you might as well get a job. If you're interested in engineering, that's totally fine. That's up to you. But if you haven't made up your mind, then try giving research a chance. If you don't know where to begin, don't worry. I'll be giving you a brief introduction into the ways of getting into colleges like Iisc Pune, Iisc Kolkata, Iisc, and NISA, which is in Orissa, if I'm not wrong. There are tons of institutes across India that provide education that is research oriented. There are 7 IISers. I'm not going to be naming them because I don't think I know all of them. There is one NISA and there is one IISc, and there are a lot more niche institutes that focus on very specific topics. As far as I've seen, there are institutes that focus on certain aspects of agricultural research. There are institutes that focus on only astronomy, and there are also institutes that focus on economic research. There are enough options and institutes out there. You just have to look for them. To give you an idea of how good these institutes are, let's look at the world ranking. IISc Pune is ranked 784th in the world, and IISc Kolkata is ranked 861st. While IISc is ranked in the top tier, it goes into the 2 digits. It's ranked at 91 to 100 category. And to give you a comparison of how IIT is ranked with respect to this, IIT is ranked around 200 and 300s. So I admit that IISers are below the IIT marks, but it doesn't necessarily mean that they're any worse. Like IITs aren't exactly all that better. Since I'm familiar with the ways to get into IISers, NICER, and IISc, I'll be talking about them. There are mainly 3 ways to get into IISers and 2 ways to get into IISc. There's JE and JE Advanced. There's KVPY. Both of these gets you into IISers and IIScs. The third one is specific only to the IISers, and it is called the IISer Aptitude Test, or IAT for short. JE and JE Advanced are pretty clear. The higher the rank you get, the easier it is for you to get into a college of your choice. As far as I know, as long as you get a JE Advanced rank of below 10,000, you'll get into one of the IISers. It might not be in your first round, but you have a chance of getting into the IISers. KVPY is one of the lesser known ways of getting into an institute. You can take this test in your 11th, 12th, and even the first year of your undergraduate. I did not know about this test until my 12th, and therefore I missed out on a very potent opportunity to get into IISer, and therefore had to repeat a whole year. This KVPY is usually held in November. Well, it was held in November when I was giving the exam, but I don't know about the settings due to the pandemic, how the settings have changed due to the pandemic. The last but unarguably the easiest way to get into an IISer is the IAT. It takes place after JEE Advanced, and the syllabus is same as that of the JEE. The level of questions, though, are far below the JEE Advanced level. They're like JEE Mains level. This is what makes it the way to go into IISers. And IISers make sure that 50% of the students in the new batch are selected through IAT, 25% through JEE Advanced, and 25% through KVPY. The other beautiful thing about studying in IISers is, and this might tempt a lot of you, is you're basically studying for free. Students who get in through KVPY are automatically eligible for a scholarship. you get around 80,000 a year. You get around 80,000 a year, but like that isn't much, is what you might be thinking right now. The thing is, the tuition fees in ICERs, IIScs, and NICER is around 30 to 35,000 a semester, while while IITs go up to 1 lakh, BITS Pilani goes up to 2 lakhs, and VIT, Vellore Institute, goes up to 4, if I'm not wrong. So the beautiful part of this is you get to study for free. Students who get in through JEE Advanced or IIT can either apply for another program called Inspire, or they can give the KVPA exam in the first year of their undergraduates. Inspire is pretty simple. It just looks for your board exams marks, and the higher you are ranked, the more chance you have of getting Inspire. KVPY, though, in the first year of your undergraduate, you're basically giving the same exam that you gave in the 12th. Like the 12th graders give the same exam as the first year undergraduates. So you can pretty much go into the exam hall unprepared and expect to get a scholarship. NICERS, and the National Institute of Science, Education and Research there is only one branch, and I believe it's in Orissa. I'm not exactly sure. It's like what NIT is to IIT. It's like what NIT is to an IIT. Like NICER is to ICER, same as NIT is to IITs. It's not bad. It's a wonderful institute, and you can definitely make strides in the field of research if you go there. But there are other options that you can look into. And to get into NICER, you cannot get into any of the 3 previous ways I've mentioned. You have to give one of their tests, which is called NEST. And I personally find it harder than the ICER aptitude test. So it's a bit more work for not so much results. The last but definitely not the least is the IISc. It requires you to get top ranks in both KVPI and JEE Advanced. And when I say top rank, I mean like top 200 in JEE Advanced and top 200 in KVPI. It is harder, but if you get into it, you get into it. It's worth it. Getting into an IISc is definitely worth it. The scholarship programs remain the same. KVPI, you get 80,000 a year. Inspire, you get 80,000 a year, and you are eligible for it the same way as that for ICERs. In short, research is the new trend. ICERs, IICs, NICERs are the places to go. The tuition fee is less than half of what the other colleges ask for. You get scholarship that you wouldn't have in IITs. And I've just given you a very brief intro into this. There are many videos that elaborately explain and break down the processes and the exams involved in getting into ICERs and IICs. But I wouldn't be going into that. You can look into those videos in your free time. And when it comes to synthetic biology, the world needs it. Like anything that is bio-based is seen as a win because of how the world is going, like how the environment is going. We need people who can work in synthetic biology. We need people who can bring about strides. We need people who can bring about changes in the world that could potentially extend humanity's lifetime on this planet. This concludes my presentation, and thank you for having me here. And if you have any questions, I'll be taking them right now.
Excellent presentation, Surya Narayan.
Thank you.
Yours. And those are doubts, any clarification, you can ask him. This is time for interaction.
Hello.
Hello.
Surya Narayan, it was a nice thing, but being too synthetic, the entire thing has not gone into the system. But I think I have made 1 or 2 I've written something, and I would like to clarify on those points. First of all, you have mentioned this when some organization or institution or factories, if they're going for that, they'll get carbon credit points, and the CSR funds can be utilized for that. That will definitely be a good thing for the organizations.
Yeah.
But as an individual, you have mentioned about natural rubber and synthetic rubber,
Yes.
and in some cases, the warnings they give is this synthetic rubber cannot be used for certain items, one, two, three.
Oh,
Well,
that is, yeah. So that is
yes,
always there. Like,
I'll complete my.
yeah, yeah. Sure, sure.
And you have mentioned about this E. coli,
Yeah, E.
but
coli.
what we hear in Alleppey is that the Vembanad Lake is having more than the allowed percentage of E. coli, which harm the human life per se. Uh, and also, my fear is about the synthetic food flavors. One may say it is bio, but it is synthetic bio. So it could harm the internal system of the people who use it also. I would like to know something more on that.
So there is this general notion that anything that is synthetic is bad. Like everyone knows about GM plants, the seedless variants that you see everywhere, and how you're not supposed to have that. That is always there, and there are cases where synthetic biology has failed. I admit that, but uh, majority of the scenarios nowadays, and because of how the FDA makes sure everything is safe when it comes to food and agriculture and all that. The FDA for US and the respective organizations for other countries, the chances that you run into a synthetic biology based compound that could potentially harm you is very less. And if it does harm you, it could be more of a personal thing, or it could also be what people call placebo. Uh, the notion that I think this will happen and therefore it happens, that kind of thing. So that could also be there. And since it is ingrained into our cells that uh, anything that is synthetic, anything that is made by manipulating genes is bad, we'll always have that. Oh no, it is synthetic, it is bad in our gene. In like that is the first thought that will come to us, and that could potentially affect the way we see all the new things that come about, and all and they could also have a physiological impact because of how you think. All I can say is when it comes to synthetic biology as food preservatives and the case of synthetic and natural rubber, I do admit like for some things you need natural rubber over synthetic rubber because synthetic rubber is sturdier and stronger, and you don't need that in all the cases. So natural rubber has its pros, it has its uses. It cannot be completely replaced. What synthetic biology does is the way synthetic rubber is produced, you have to burn petrochemicals. You need natural rubber as a raw material, and you have to burn petrochemicals to produce synthetic rubber. But the way synthetic biology works is you can either take the same natural rubber from the trees, or you can produce the same natural rubber in a bacteria and then make it synthetic. Like the whole process that takes place in the petrochemical industry can be simulated in the bacteria itself. So we're not technically uh, overthrowing rubber. We need natural rubber. We need that compound. The existence of natural rubber still has its importance. The only thing that we are trying to overthrow is a petrochemical aspect of it. We are trying to reduce the carbon emissions from the petrochemical manufacturing, not take out rubber as a whole, not replace natural rubber with synthetic rubber. Natural rubber has its uses. Like for some things you need natural rubber, you can't use synthetic rubber, it's too strong. So there are uses for natural rubber. And when it comes to and petrochemical, like the main target of any bio manufacturing is to reduce the petrochemical manufacturing of the same, so that the carbon emissions are reduced. And obviously, like any industry, the carbon emission is reduced means they get more carbon credit, and they get more spare carbon credit. They don't get more absolute carbon credit. They get more spare carbon credit, and it extends the humanity's stay in this planet. And like I said, by 2030, we need to reduce our carbon emissions by 45%. It's like by half. If you cut down the manufacturing of everything by half, we could reach that. We have reached the point where just planting trees cannot save our planet anymore. So we need ways in which you can reduce the emissions along with planting more trees. So all I would say is, when it comes to synthetic biology, have an open mind, like try to be more accepting of it. And there are cases where they fail, try not to focus on them because that is the best we can do. It's good both for our mental and physical health. Like we won't be thinking anything negative of something new, and also we would be able to enjoy the creations that our future generations are providing to us. And as I said, for natural rubber and synthetic rubber, they have their own pros and cons. The only thing that synthetic biology is trying to do is reduce the petrochemical aspect of it and not replace synthetic rubber and natural rubber with each other.
Yes, it's true. The research people and the research institutions will have to address this as a thing, and the people, the on the street, they also should understand.
Yeah, so that is the point of our HP, like how we taught students about climate change, how we taught students about synthetic biology, like the YouTube video series that we've made it in such a way that you can mute the video and see, and you will still understand everything because everything is drawn from scratch. You can see how everything is formed. You understand everything just by the visuals itself. So you don't need to listen to the technical terms that is being said in the audio. You can literally mute the video and still understand everything. And as we mentioned, there was audio webinar for people who cannot visually who cannot see stuff. There was a silent movie for people who cannot listen, like who are hard of hearing. There was also art competitions for people who are engaged in art. So in a way, we are trying to bring about synthetic biology, its uses, and the adverse state our world is going into through things that people themselves find interesting, like art for people who do art. We had poster making competition, art competition. People who can't see, they had an audio webinar like this, but like there were no visuals, there was no presentation. It was just two people saying narrating a story, and the story had all the content. And there was also a silent film for people who can't hear. So we are trying our best to include everyone on the street to understand what synthetic biology is. And IGEM wants people to IGEM wants each of their teams to do enough HP so that they can get a medal. So imagine there are 353 teams. That was in 2019. As of this year, I believe the numbers were more. It reached around almost 400. And there are 400 teams. And each of the teams, let's say there's an average of 10 students. That makes 4,000 students. 4,000 students at an undergraduate, graduate, and high school level are putting an effort to bring people from the streets into synthetic biology, like bringing them to the point where they can accept the efforts, the results of synthetic biology, and also alert them about all the various local and global issues that are there, like that you wouldn't find in a newspaper. Like in a newspaper, what the maximum you would say is this person killed that person, there was a scandal. You're not going to see, oh no, the world is coming to an end because of everything that we humans are doing. So every single team that participates in IGEM puts in enough amount of effort to get every common person involved in all the science, all the synthetic biology, and everything else. So we are doing our best, and obviously the competition hosts, the iGEM themselves have their own programs where they do this outreach stuff, where they teach people about synthetic biology and everything else.
Thank you.
Some question is there. Some messages are there from please.
As I said, I'm a physics major, so I'm not exactly sure about the branches of biology. But as far as I know, genetics is about you studying genetics. Like, say you're doing a genome sequencing, anything like that. That is genetics. Understanding how genetics works, like understanding how genes are affected, and to some extent, as far as I know, like my classmates have taken genetic courses, and they just learned about the genetic variations that came from generations of animals, um, like as time passed. And but synthetic biology works with when you start engineering on the when you start engineering, like literally become an engineer with the knowledge that you have from all the genetics, you become a synthetic biologist. That is the simplest way of putting it. You start breaking things down, you start putting things together, you start cutting things here, pasting it there, you start whatever comes to your mind with the genetics. Instead of just sitting there looking at it, studying, seeing the pattern, you become a synthetic biologist. Does that answer your question?
Anybody else? ആരെങ്കിലും സംസാരിക്കാൻ? ഡൗട്ട്? ഡൗട്ട് വല്ലതും ഉണ്ടോ? വളരെ നല്ല പ്രസന്റേഷൻ ആയിരുന്നു.
Thank you.
ബയോളജിയെ കുറിച്ചുള്ള വസ്തുത വളരെ ഡീറ്റെയിൽ, വളരെ നല്ലൊരു അറിവാണ്. പെട്രോ കെമിക്കൽസിന്റെ ആ ഒരു പ്രശ്നങ്ങൾ വലിയൊരു ചലഞ്ചാണ് വാസ്തവത്തിൽ.
അതെ, കാരണം നമ്മളിപ്പോ പെട്രോ കെമിക്കൽസിൽ എത്ര ഡിപ്പെൻഡന്റ് നോക്കൂ. എന്താ, കാർ ഓടിക്കണ പെട്രോ കെമിക്കൽ വേണം, തീ കത്തിക്കണമെങ്കിൽ LPG വേണം, എല്ലാത്തിനും പെട്രോ കെമിക്കലിന്റെ ആവശ്യമുണ്ട്. അപ്പോ അങ്ങനെ വരുന്ന ഒരു ഇതില് ഈ ബയോഫ്യൂവൽസ് ഇപ്പൊ തന്നെ എന്താ വീട്ടിൽ നിന്ന് പറഞ്ഞ ഭക്ഷണ വേസ്റ്റുകൾ ഉപയോഗിച്ചിട്ട് ഇത് ഗ്യാസ് ഉണ്ടാക്കൽ ഒരു വലിയ സംഭവമായിട്ട് മാറിയല്ലോ. അപ്പൊ അതേപോലെ സിന്തറ്റിക് ബയോളജി ആകുമ്പോൾ കുറച്ചും കൂടി എഫിഷ്യന്റ് ആയിരിക്കും, കുറെ കൂടെ പ്രോഡക്റ്റ് കിട്ടും. അത്രേ ഉള്ളൂ വ്യത്യാസം.
പൊല്യൂഷൻ പറഞ്ഞിട്ടും കാർബൺ
അതെ അതെ
ഡൽഹിയിലൊക്കെ അനുഭവിക്കുന്ന ഏറ്റവും വലിയ പ്രശ്നം പൊല്യൂഷനാണ്.
അതെ
വലിയ പ്രശ്നം. അതുപോലെ കൃത്യം പ്ലാന്റ്സിനെ ബാധിക്കും, നമ്മുടെ ഫുഡ് സൈക്കിളിനെ ബാധിക്കും.
അതെ തന്നെ. ഫുഡ് മാത്രല്ലല്ലോ, ഇപ്പൊ തന്നെ അറിയാലോ, ഇപ്പൊ ചൂട് എന്ന് പറഞ്ഞാൽ ഭയങ്കര ചൂടാണ്, തണുപ്പ് കാലത്താണ് തണുപ്പും ഇല്ല, മഴക്കാലത്താണ് മഴ എങ്ങനെയാ വരുന്നത് ആരും പറയാൻ പോലും പറ്റില്ല. അപ്പൊ അങ്ങനത്തെ ഇതൊക്കെ വരുന്നത് ഇങ്ങനെ കാർബൺ ഡൈ ഓക്സൈഡിന്റെ ഇത് കൂടുമ്പോഴാണ്.
നല്ലൊരു സബ്ജക്റ്റ് ആണ് വാസ്തവത്തില്. ഭാവിയിലെ വളരെ മുന്നിൽ കാണുന്നത്. ഭാവി കാലത്ത് ഏറ്റവും അധികം ജനങ്ങൾക്ക് സഹായകം
പിന്നെ ബയോ പീറ്റ് മോസ് എല്ലാം യൂസ് ചെയ്യുന്നത് ആക്ച്വലി അതെല്ലാം വളരെ നല്ല കാര്യമാണ്.
അതന്നെ കുറെ ഉണ്ട്. ഇപ്പൊ കുറെ ഈ ഗ്രീൻ വേസ് ഓഫ് പ്രൊഡ്യൂസിങ് എന്ന് പറഞ്ഞിട്ട് കുറെ ഉണ്ട്. അത് നാടൻ കുറെ മെത്തേഡ്സ് ഉണ്ട്, അത് എല്ലായിടത്തും അറിയാത്ത കുറെ മെത്തേഡ്സ് ഉണ്ട്. സിന്തറ്റിക് ബയോളജി എന്ന് പറയുന്നത് കുറച്ചും കൂടെ സയൻസ് ഇതാണ്. അതായത് കാരണം ഒരു ലെവൽ ഓഫ് സ്കിൽ ഇല്ലാണ്ട് ഇത് ചെയ്യാൻ പറ്റില്ല. കാരണം ജീൻസ് ഒക്കെ എഡിറ്റ് ചെയ്യാന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ അതിനൊരു ലെവൽ ഓഫ് സ്കിൽ വേണം. ഈ ഇത് സിന്തറ്റിക് ബയോളജി അല്ലാണ്ട് കുറെ ഈ നാടൻ കുറെ സൂത്രങ്ങളുണ്ടല്ലോ, അതൊക്കെ യൂസ് ചെയ്യാം. അതും സെയിം റിസൾട്ട് തന്നെയാ തരണേ, അതില് വ്യത്യാസമൊന്നുമില്ല. നമുക്ക് നമ്മളുടെ ഇത് എത്ര ചെറിയ ഹെൽപ്പ് കിട്ടുമോ അത് വേണം ഇത് മുന്നോട്ട് കൊണ്ടുപോകണമെങ്കിൽ, പ്ലാനറ്റ് മുന്നോട്ട് കൊണ്ടുപോകണമെങ്കിൽ. അപ്പൊ
പെട്രോൾ ഫ്രം പായൽ, രാമർ പെട്രോൾ.
അത് തന്നെ അങ്ങനത്തെ കുറെ സാധനങ്ങളുണ്ട്.
മെഡിസിനൽ ഇമ്പോർട്ടൻസ് ഉണ്ടോ? മെഡിസിനൊക്കെ പറ്റുമോ? അങ്ങനെ ഉണ്ടല്ലേ. നമുക്കിപ്പം ഈ നമ്മുടെ ആൻസർ നമ്മൾ ഇതായിട്ട് നമ്മൾ പറയാനില്ല, അങ്ങനെയൊക്കെ നമുക്ക് പാരമ്പര്യമായിട്ടുണ്ടെന്ന് പറയുന്നു. അപ്പൊ ഈ രീതിയിൽ ബയോളജിയുടെ ഈ പറഞ്ഞ ട്രാൻസ്ഫോർമേഷൻ ഉപയോഗിച്ച് അങ്ങനെയുള്ള മരുന്നുകളൊക്കെ വേണമെങ്കിൽ ഈ ജീൻസ് ചേഞ്ച് ചെയ്യാൻ വേണ്ടി ഉപയോഗിക്കാൻ പറ്റൂലേ? അങ്ങനെ വരുമ്പോൾ അങ്ങനെ പറ്റുമോ?
അപ്പോ അതാണ് ഞങ്ങളുടെ മുമ്പത്തെ ടീം എന്ന് പറയുന്നത്. ഞങ്ങളുടെ മുമ്പത്തെ ടീം 2020 ടീം, അവര് ചെയ്തത് അവരൊരു ഡയഗ്നോസ്റ്റിക് കിറ്റ് ഉണ്ടാക്കി. അതായത് ഈ നിങ്ങൾ ഹോസ്പിറ്റലിൽ പോയി ടെസ്റ്റ് ചെയ്യുമല്ലോ, ബ്ലഡ് കൊടുത്തിട്ട് ഇത് ടെസ്റ്റ് ചെയ്യുമല്ലോ ഏതൊക്കെ അസുഖം ഉണ്ടെന്ന് അറിയാൻ, അതേപോലത്തെ ഒരു സാധനമാണ്. അവര് ഒരു ഡയഗ്നോസ്റ്റിക് കിറ്റ് ഉണ്ടാക്കി. ആ കിറ്റില് അവര് എഞ്ചിനീയർ ചെയ്ത ബാക്ടീരിയ ആണ് അതിൻ്റെ അകത്തുള്ളത്. അപ്പൊ അവര് പിന്നെ മെഷീൻ ലേണിംഗ് എന്ന് പറഞ്ഞിട്ടൊരു സംഭവം ഉണ്ട്. എന്താ കമ്പ്യൂട്ടർ വെച്ചിട്ട് പ്രഡിക്റ്റ് ചെയ്യണതാണ് അത്. ഇതുവരെ എങ്ങനെയാണോ സംഭവം നടന്നേക്കണേ അത് നമ്മൾ കമ്പ്യൂട്ടറിന് കൊടുത്തു കഴിഞ്ഞാൽ കമ്പ്യൂട്ടർ പറയും ഈ സാമ്പിളില് ഫ്യൂച്ചറിൽ എന്തൊക്കെ നടക്കാൻ പറ്റും, ഇതൊക്കെയാണ് പോസിബിലിറ്റി, ഇത്രയും പെർസെന്റേജ് ഇത് നടക്കും, ഇത്രയും പെർസെന്റേജ് ഇത് നടക്കും. അതാണ് മെഷീൻ ലേണിംഗ് എന്ന് പറയുന്നത്. അപ്പോ അവർ ഈ മെഷീൻ ആർട്ടിഫിഷ്യൽ ഇന്റലിജൻസ്, അപ്പൊ ആർട്ടിഫിഷ്യൽ ഇന്റലിജൻസും ഈ സിന്തറ്റിക് ബയോളജി ഒരു ബാക്ടീരിയയിൽ യൂസ് ചെയ്തു. എന്നിട്ട് ആർട്ടിഫിഷ്യൽ ഇന്റലിജൻസ് ആ ബാക്ടീരിയയിലെ ആ ബാക്ടീരിയ ഒരു സാധനം പ്രൊഡ്യൂസ് ചെയ്യും. ഒരു അസുഖത്തിന് ഒരു എ കോമ്പൗണ്ട് പ്രൊഡ്യൂസ് ചെയ്യുകയാണെങ്കിൽ വേറൊരു അസുഖത്തിന് ബി കോമ്പൗണ്ട് പ്രൊഡ്യൂസ് ചെയ്യും. അപ്പൊ അങ്ങനത്തെ കോമ്പൗണ്ട്സ് പ്രൊഡ്യൂസ് ചെയ്യല് ആ ബാക്ടീരിയനെ അവര് സിന്തറ്റിക് ബയോളജി വഴി കേപ്പബിൾ ആക്കി. അപ്പൊ അങ്ങനെ ആക്കിയിട്ട് ഇവർ ഈ AI ഉപയോഗിച്ചിട്ട് ഒരാൾക്ക് എന്ത് അസുഖം ഉണ്ടാവും എന്ന് പ്രെഡിക്റ്റ് ചെയ്യാനായിട്ടുള്ള ഒരു ഡയഗ്നോസ്റ്റിക് കിറ്റാണ് ഉണ്ടാക്കിയിരിക്കുന്നത്. അവരത് ഈ വർഷം അവരൊരു സ്റ്റാർട്ടപ്പ് തുടങ്ങിയിട്ട് അവർ ഈ IGEMന്റെ പ്രോഗ്രാമില് പിച്ച് ചെയ്തു, സ്പോൺസർഷിപ്പ് പിച്ച് ചെയ്തു. എന്നിട്ട് അവർക്ക് $10,000 സ്പോൺസർഷിപ്പും കിട്ടി. അവരൊരു ഒഫീഷ്യൽ സ്റ്റാർട്ടപ്പായി മാറിയിട്ടുണ്ട്.
ഇത് ഇപ്പോഴത്തെ പുതിയ ഈ പേഴ്സണൽ ജിനോമിക്സ് എന്ന് പറയുന്നതും ഇതിന്റെ ഒരു ഭാഗമായിട്ട് ഒന്നതായിരിക്കണം അല്ലേ?
പേഴ്സണൽ ജീനോമിക്സ് എന്ന് പറയുന്നത്
ഈ ഹ്യൂമൻ DNA നോക്കി കഴിഞ്ഞിട്ട് നമുക്ക് എന്താ വരാം.
അതന്നെ അത് ഞാൻ ആ ഡയബറ്റീസിന്റെ എക്സാമ്പിൾ പറഞ്ഞ പോലെ തന്നെ. അതായത് അച്ഛനും അമ്മയ്ക്കും ഉണ്ടെങ്കിൽ തനിക്ക് കിട്ടാൻ ചാൻസ് ഉണ്ട് എന്ന് പറയണ പോലെ. പക്ഷെ എന്ന് പറഞ്ഞിട്ട് അച്ഛനും അമ്മയ്ക്കും ഉണ്ട് എനിക്ക് കിട്ടും എന്ന് പറഞ്ഞിരുന്ന ആവശ്യമുള്ള മധുരമൊക്കെ ഇപ്പൊ കഴിക്കാൻ പറ്റുന്ന സമയത്ത് കഴിച്ചു എന്ന് പറഞ്ഞാൽ കാര്യമില്ലല്ലോ.
അപ്പൊ ഈ കൊറോണയുടെ കേസിൽ ചൈനക്കാർ ഈ സബ്ജക്റ്റ് ആണ് ഉപയോഗിച്ചേക്കുന്നതെങ്കിൽ നമുക്ക്
മിക്കവാറും. കാരണം ഞാനിപ്പോ പറഞ്ഞത് ബാക്ടീരിയയുടെ കുറിച്ച് മാത്രമാണ്. വൈറസസിന്റെ കാര്യം എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ അവരെ ഒരു ജീവിയായിട്ട് കാണാൻ പറ്റില്ല. അവർക്ക് ലൈഫ് ഇല്ല. ലൈഫ് ഉള്ളത് ഒരു ഹോസ്റ്റ്, അതായത് ഒന്നില്ല മനുഷ്യരോ അല്ലെ പ്ലാന്റ്സോ അല്ലെ ബാക്ടീരിയയുടെ ഉള്ളിൽ കയറുമ്പോഴാണ് അവര് ലൈഫ് ആകാൻ പോണത്. അതുവരെ ഞാൻ ഈ പറഞ്ഞ പോലെ ഇപ്പൊ DNA ചുമ്മാ എടുത്തു. DNA എന്ന് പറഞ്ഞാൽ എന്താ, കുറെ കെമിക്കൽ കോമ്പൗണ്ട്സ് ഒരുമിച്ചിരിക്കണതാ. അതെടുത്ത് ഞാൻ ഇപ്പൊ നിലത്തിട്ട് ഞാൻ ജീവിക്കാൻ പോണില്ലല്ലോ. അതാണ് വൈറസ് എന്ന് പറയുന്ന ഒരു സാധനം. വൈറസ് എന്ന് പറയുന്നത് അങ്ങനത്തെ ഒരു കോമ്പൗണ്ട് എടുത്ത് ചുമ്മാ നിലത്തിട്ട് അങ്ങനെ ഇരിക്കും, അതാണ്. പക്ഷെ ആ കോമ്പൗണ്ട് എടുത്ത് ഒരു ജീവിയുടെ ഉള്ളിലോട്ട് ഇടുകയാണെങ്കിൽ അത് എന്ത് ചെയ്യുമെന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ ആ ജീവിയുടെ ജീൻ സെറ്റ് ഉണ്ടല്ലോ, ജീനോം ഉണ്ടല്ലോ, ആ ജീവിയുടെ ജീൻസിന് ഉള്ളിലോട്ടേക്ക് അത് കയറിപ്പെട്ട് കഴിഞ്ഞാൽ പിന്നെ അത് ജീവിയുടെ കേപ്പബിലിറ്റീസ് വെച്ചിട്ട് ജീവിക്കാൻ തുടങ്ങും. അതായത് അതിന് ആവശ്യമുള്ള എന്താണോ അതെല്ലാം പ്രൊഡ്യൂസ് ചെയ്യാൻ തുടങ്ങും. അതാണ് വൈറസ് വരുന്നത്. അപ്പൊ വൈറസ് അങ്ങനെ, അപ്പൊ അതും ഈ വൈറസ് എന്ന് പറയുന്നതും ഞാൻ പറഞ്ഞല്ലോ ആ DNA താഴെ ഇട്ടു എന്ന് പറഞ്ഞാൽ അത് ജീവിക്കാൻ പോണില്ല. അപ്പൊ DNA സീക്വൻസ് ആണ് വൈറസ് എന്ന് പറയണത്. അപ്പൊ സിന്തറ്റിക് ബയോളജിയിൽ സീക്വൻസ് ഉണ്ടാക്കാൻ പറ്റുമെങ്കിൽ വൈറസ് ഉണ്ടാക്കാൻ പറ്റുന്നത് വലിയ കാര്യമില്ല. പക്ഷെ വൈറസ് എന്ന് പറഞ്ഞാൽ വേറൊരു സംഗതിയാണ്. DNA അല്ല, RNA എന്ന് പറഞ്ഞിട്ടൊരു സംഭവമാണ്. അത് വ്യത്യാസം എന്ന് പറഞ്ഞാൽ RNA കുറച്ചും കൂടെ അൺസ്റ്റേബിൾ ആണ്, പെട്ടെന്ന് പൊട്ടിപ്പോവും. DNA പെട്ടെന്ന് പൊട്ടില്ല. പിന്നെ DNA ഇൻഫർമേഷൻ സ്റ്റോർ ചെയ്യാൻ വേണ്ടിയിട്ട് യൂസ് ചെയ്യണതാണ്. RNA കൂടുതലൊരു ഇതുപോലെ ഒരു ഇക്വേഷൻ പോലെ, ഒരു കെമിക്കൽ ഇക്വേഷൻ പോലെയാണ്. ഇതുണ്ടെങ്കിൽ ഇതുണ്ടാക്കുക, എന്നെ റീഡ് ചെയ്യുകയാണെങ്കിൽ ഇതുണ്ടാക്കുക. അതാണ് RNA എന്ന് പറയുന്നത്. അപ്പൊ RNA ഈ വൈറസ് ഒരു ഹോസ്റ്റ് കയറിപ്പെട്ട് കഴിഞ്ഞിട്ട് അതിന്റെ ജീനോമില് കയറിപ്പെട്ട് കഴിഞ്ഞാൽ അതിനെ റീഡ് ചെയ്യുമ്പോഴത്തേക്കും ഇതുണ്ടാക്കാൻ പറയും. അപ്പൊ ഈ ഇതെന്ന് പറയണത് കുറെ കൂടെ വൈറസ് ആയിരിക്കും. അപ്പോ അങ്ങനെ വൈറസിന്റെ എണ്ണം ഒരു ജീവിയിൽ കൂടിക്കൊണ്ടിരിക്കും. അവസാനം വൈറസ് മാത്രമേ ഉണ്ടാവുള്ളൂ. പിന്നെ വൈറസ് അതിൽ നിന്ന് പുറത്തോട്ട് ചാടാൻ തുടങ്ങും. അത് അങ്ങനെയാണ് കൊറോണയൊക്കെ പടരുന്നത്. കാരണം തുമ്മിക്കഴിഞ്ഞാൽ എന്താ ചിലപ്പോൾ നിങ്ങളുടെ മ്യൂക്കസിലുണ്ടാവും, അതെടുത്ത് പുറത്തു വന്നു. ഇനി ആ വൈറസ് കാറ്റത്തൂടെ പറന്ന് എല്ലായിടത്തും പോവും. അതാണല്ലോ കൊറോണയ്ക്ക് നടന്നത്. അപ്പൊ അങ്ങനത്തെ ഇതൊക്കെ ഉണ്ട്. ഇതിന്റെ എന്താ ബയോ ഹാസാർഡ് രീതിയിലുള്ള ഒരു സൈഡും ഉണ്ട്, നല്ല സൈഡും ഉണ്ട്. കുട്ടികൾ എപ്പോഴും നല്ല ഇതല്ലേ ഫോക്കസ് ചെയ്യുക. കാരണം ഞങ്ങൾ എന്തായാലും ആരും ഞങ്ങൾക്ക് കാശ് തരില്ലല്ലോ ഒരു ബയോ വെപ്പൺ ഉണ്ടാക്കാൻ. അപ്പൊ പിന്നെ അസുഖം കാര്യമില്ല, അത് ഒരു ഗുണമില്ല.
ആസ് എ സ്റ്റുഡന്റ് യു ഹാവ് ജസ്റ്റിഫൈഡ് ഓൾസോ. ഈ നാച്ചുറൽ റബ്ബർ സിന്തറ്റിക് റബ്ബറിനെ കുറിച്ച് റിപ്ലൈ ചെയ്തപ്പോഴത്തേക്കും തന്നെ പറഞ്ഞത് സിന്തറ്റിക് റബ്ബർ ഈസ് മോർ സ്ട്രോങ്. അത് ഐ വാസ് ആക്ച്വലി ഹോപ്പിംഗ് ദി അതർ വേ. ഈ നമ്മൾ സാധാരണ പറയും പെട്രോൾ പമ്പിൽ യൂസ് ചെയ്യുന്ന ഹോസ് യു കനോട്ട് യൂസ് എ സിന്തറ്റിക് റബ്ബർ, യു കൻ ഓൺലി യൂസ് എ നാച്ചുറൽ റബ്ബർ. എൻ്റെ ടെക്നിക്കൽ സൈഡ് എനിക്ക് അറിഞ്ഞുകൂടാ. പക്ഷേ അത് ഞാൻ ഉദ്ദേശിക്കുന്നത് സിന്തറ്റിക് റബ്ബറിന് കൂടുതൽ സ്ട്രോങ് ആണ് എന്നല്ല. സിന്തറ്റിക് റബ്ബർ ഈസ് മോർ നാച്ചുറൽ റബ്ബർ ഈസ് മോർ സ്ട്രോങ് ഇൻ ദ സെൻസ് ബെറ്റർ എന്നാണ് നമ്മുടെയൊക്കെ മനസ്സിൽ കയറിയിരിക്കുന്നത്.
ശരിയാ. പക്ഷെ ഇപ്പൊ പെട്രോൾ പമ്പിലെ ഹോസിന്റെ കാര്യം പറയുമ്പോ അത് അതേ മെറ്റീരിയൽ ഇപ്പൊ ടയറിന് ഉപയോഗിച്ച് കഴിഞ്ഞാൽ എന്ത് പറ്റും? ഈ റോഡിൽ കൂടെ പോണ ടയർ, ഇത്രയും സ്പീഡിൽ പോണ ടയറിന്. അപ്പൊ അങ്ങനെയൊക്കെ വരുമ്പോ കുറച്ചും കൂടെ ഒരു സ്ട്രെങ്ത് വേണം ആ മെറ്റീരിയലിന്. അപ്പൊ സിന്തറ്റിക് റബ്ബർ ഇവര് തുടങ്ങി വെച്ചത്, ഇവര് ഉണ്ടാക്കിയത് തന്നെ അതിന്റെ എന്താ സ്ട്രക്ചറൽ ഇന്റഗ്രിറ്റി ഇൻക്രീസ് ചെയ്തുകൊണ്ടാണ് ഉണ്ടാക്കിയത് തന്നെ. അപ്പോ അങ്ങനത്തെ രണ്ടും ആവശ്യമുണ്ട്. ഇപ്പൊ സിന്തറ്റിക് റബ്ബർ ഇവിടെ എന്താ പെട്രോൾ പമ്പിൽ ഓസിൽ യൂസ് ചെയ്യാൻ പറ്റില്ല എന്ന് പറഞ്ഞ പോലെ ചില സ്ഥലത്ത് നാച്ചുറൽ റബ്ബർ യൂസ് ചെയ്യാൻ പറ്റില്ല, സിന്തറ്റിക് വേണം. അല്ലാതെ ഒരെണ്ണം മറ്റേനേക്കാളും നല്ലതെന്നല്ല പറയണ.
ഞാൻ പറയുന്നത് ഇറ്റ് ഷുഡ് ബി ഈക്വൽ. ആ ഒരേ സ്റ്റാൻഡേർഡിലേക്ക് കൊണ്ടുവരാൻ സാധിക്കണം. ഇറ്റ് ഷുഡ് ബി
അതായത്
സബ്സ്റ്റിറ്റ്യൂട്ട്.
അതിന് രണ്ടിനും അവരവരുടെ യൂസ് ഉണ്ട്. പക്ഷെ ചിലതിന്റെ യൂസിൽ സ്ട്രെങ്ത് കൂടുതൽ ഒരു എണ്ണം വേണമെന്ന് പറയുമ്പോഴത്തേക്കും സിന്തറ്റിക് റബ്ബർ ഉപയോഗിക്കണമല്ലോ, അത്രേ ഉള്ളൂ.
അല്ല, നമ്മൾ ഒരു സാധനം ഡെവലപ്പ് ചെയ്യുമ്പോഴത്തേക്കും അതും സയന്റിഫിക്കലി വിത്ത് ലോട്ട് ഓഫ് എഫേർട്സ് ചെയ്യുമ്പോഴത്തേക്കും വി ഷുഡ് ട്രൈ ടു മേക്ക് ഇറ്റ് ആസ് പ്രിസൈസ് ആസ് പോസിബിൾ, അല്ലേ? ടു മേക്ക് ഇറ്റ് ഇൻ യൂണിസൺ, രണ്ടും ഒരേപോലെ ആവാൻ വേണ്ടിയല്ലേ ശ്രമിക്കേണ്ടത്.
അല്ല, പക്ഷെ അതാണ് സിന്തറ്റിക് റബ്ബർ എന്ന് പറയുന്നത് കെമിക്കലി ഡിഫറെന്റ് ആണ് നാച്ചുറൽ റബ്ബറിനെക്കാളും. അത് കാരണമാണ് അത് സ്ട്രോങ് ആയിട്ട് വരുന്നത്. അതായത് നാച്ചുറൽ റബ്ബർ ഉപയോഗിച്ചിട്ടാണ് സിന്തറ്റിക് റബ്ബർ ഉണ്ടാക്കുക. നാച്ചുറൽ റബ്ബറിലോട്ട് അവർ സൾഫർ ഇട്ട് ഹീറ്റ് ചെയ്ത് എന്തൊക്കെ ചെയ്യുമ്പോഴാണ് സിന്തറ്റിക് റബ്ബർ വരുന്നത്. അതായത് സിന്തറ്റിക് റബ്ബറിന്റെ കോമ്പോസിഷൻ തന്നെ വ്യത്യാസമുണ്ട്. എനിക്ക് കെമിസ്ട്രി അറിയില്ല അതിന്റെ. എന്റെ സ്കൂൾ പഠിത്തത്തിന്റെ അറിവില് അവര് സൾഫർ ഇട്ടിട്ട് ഈ നാച്ചുറൽ റബ്ബറിന് കുറെ ലെയേഴ്സ് ഉണ്ട്. അതാണ് നാച്ചുറൽ റബ്ബറിനെ ഇങ്ങനെ ഭയങ്കര വളയ്ക്കാൻ പറ്റുന്ന രീതിയിലൊക്കെ ആക്കണ. അവർ ഈ ലെയേഴ്സിനെ കണക്ട് ചെയ്യണത് സൾഫർ വെച്ചിട്ടാണ്. അപ്പൊ ആ സൾഫർ വെച്ച് അങ്ങനെ ലെയേഴ്സിനെ കണക്ട് ചെയ്യുമ്പോഴാണ് കുറച്ചും കൂടെ സ്ട്രെങ്ത് കൂടുമല്ലോ. കാരണം ഇനിയിപ്പോ വളയാൻ പറ്റില്ലല്ലോ. അങ്ങനെയാണ് സിന്തറ്റിക് റബ്ബർ ഉണ്ടാക്കിയത്. അവര് സിന്തറ്റിക് റബ്ബർ ഉണ്ടാക്കിയത് നാച്ചുറൽ റബ്ബറിനെ റെപ്ലിക്കേറ്റ് ചെയ്യാനല്ല, അതിനെ കോപ്പി ചെയ്യാനല്ല. അവരുണ്ടാക്കിയത് സിന്തറ്റിക് ഉണ്ടാക്കിയത് വേറെ പെർപ്പസസിന് വേണ്ടിയിട്ടുള്ളതാ. cheaper item that can be used in places that natural rubber cannot be used. അല്ലാതെ നാച്ചുറൽ റബ്ബർ യൂസ് ചെയ്യുന്ന സ്ഥലത്ത് നാച്ചുറൽ റബ്ബർ തന്നെ മതി എന്നാണ് അവര്.
ഈ ഗുഡിയറിന്റെ മെൻഷൻ ചെയ്തുകൊണ്ട് അവർ മാത്രമേ അത് ആ ടെക്നോളജി ഉപയോഗിച്ചുള്ളൂ. ഈ പറഞ്ഞ ഗുഡിയറിന്റെ കാര്യം പറയാൻ കാര്യം മറ്റു കമ്പനികൾ
അല്ല, അത് കാരണം അവര് പേറ്റന്റ് അവരുടെ കയ്യിലായോണ്ടാണ്. അല്ലാണ്ട് അവര് മാത്രല്ല, എല്ലാ കമ്പനീസ് എന്റെ അറിവിലല്ല, എനിക്ക് തോന്നുന്നു എല്ലാ കോർപ്പറേറ്റ് കമ്പനീസും ഇങ്ങനത്തെ ഗ്രീനർ വേസ് ഓഫ് പ്രൊഡ്യൂസിങ് കോമ്പൗണ്ട്സ് അതിനുള്ള എഫേർട്ട് ഫുൾ ആയിട്ട് ഇടുന്നുണ്ട്. കാരണം ഞാൻ പറഞ്ഞല്ലോ, CSR ഫണ്ട്സ് എല്ലാ കമ്പനി ഇപ്പൊ കൊടുക്കണം എന്നായി. അപ്പോ അങ്ങനെ വരുമ്പോഴത്തേക്കും എല്ലാ കമ്പനിക്കും ഉണ്ടാവും ഇങ്ങനത്തെ ഒരു ഇത്. ഞാൻ എന്താ ഗുഡിയർ ഒരു എക്സാമ്പിൾ ആയിട്ട് യൂസ് ചെയ്തു എന്ന് മാത്രമേ ഉള്ളൂ. വേറെ കുറെ കമ്പനീസ് യൂസ് ചെയ്യുന്നുണ്ട്, ഇതിൽ പാർട്ടിസിപ്പേറ്റ് ചെയ്യുന്നുണ്ട്.
So you have said that the cells you use, you take the DNA and you can change the characters of it.
Yeah.
Yeah. So you take the DNA from the nucleus or from the mitochondria?
We'll most probably be working with nucleus DNA, not mitochondria. Because like all the hereditary information is stored in the nucleus DNA. So anything that we need to change in appearance or anything will be taking up from the nucleus DNA, not mitochondria.
Yeah, because it's usually the mitochondria's characters usually never change.
Yeah, exactly.
While the nucleus is always changed.
Exactly. So all the variations that happen from generation to generation happens in the nuclear DNA. So that is what we would want to work with, because we need to induce variations into the organism that you're working with. So it makes sense to work with nuclear over mitochondria.
Yeah, thank you.
Wait, you're already a biotech student, no? Like second year doing your BTech and biotech, no?
Yes.
I don't know. I think with your biotech degree itself, like you are working with technology and biology, you can pretty much like instead of yeah, you don't need a science. Like so the thing is, synthetic biology is very open. You don't need a science degree per se. You can do it with a technology degree as well. So the thing is, like, as far as I know, all the people who come out of ISIS and ISCs, like who come out with a BSc degree, they don't normally just work and research. They can very well choose engineering. Like you're just a second year. You don't have to worry about what you're gonna do after your college right now. You can like finish your course. You can do all your studies, and then if you find synthetic biology to still be very interesting, you can put in the effort outside of your college, like after you're done. And if you have connections, you can like get into some R&D lab that works with synthetic biology. You don't need to give up your second year, because if you're going to give up your second year right now, waste 1 year studying for JE or any other exam, then you're like, you're taking a very big risk. You might as well just finish your second year in a biotech. And since it's biotech, you have a higher chance of working with synthetic biology. You don't need to worry about anything. Plus, I don't think you'll be allowed to take an undergraduate course, because you have to be in that certain age group if you're taking an undergraduate course. Yeah, I don't think people who are past 3 repeat years are allowed to go into undergraduate courses. So if you drop out now and take a year to study for JE or any other exam, which is the only way to get into ISIS, then you will have a problem, because the chances of you being eligible to join ISIS is very slim. So the safe thing would be to just continue with your biotech degree, finish your biotech degree, and then. Oh wait. Don't you work? Don't you have to do internships? You can do like a synthetic biology internship and make connections from there and work in the synthetic biology department after that. Or like, I'm pretty sure you have connections with your professors. You can just ask one of them to give you a contact of someone who works with synthetic biology. I'm pretty sure they'll know. Anyone working in biotech gonna work with synthetic biologists as well. Yeah, so in your internships, you can make a lot of contacts, and you like make contacts. Like the one thing I would tell you is to make as much contacts as you can, because after a point, nepotism is the only way you can get through life. If you want a job, you need to have some power, you need to have some connections within the organization. Just make contacts and like get into some synthetic biology aspects of it through the contact.
അപ്പൊ എല്ലാവരുടെയും ഡൗട്ട് കഴിഞ്ഞു എന്ന് വിശ്വസിക്കുന്നു. ഒരു നല്ല പ്രസന്റേഷൻ ആയിരുന്നു.
Thank you.
പരിപാടിയുടെ അവസാനം എന്ന നിലയ്ക്ക് ഞാൻ നന്ദി കൃതജ്ഞത പറയാനായിട്ട് ക്ഷത്രിയക്ഷേമ സഭയുടെ സംസ്ഥാന ട്രഷറർ ശ്രീ മഹേഷ് രാമവർമ്മയെ ക്ഷണിച്ചു.
എല്ലാവർക്കും നമസ്കാരം. വളരെ നന്നായിരുന്നു സൂര്യ. എൻ്റെ കർത്തവ്യത്തിലേക്ക് കടക്കുകയാണ്. സഭയുടെ ഈ പ്രോഗ്രാമെല്ലാം കോർഡിനേറ്റ് ചെയ്ത് നമ്മോടൊപ്പം എപ്പോഴും വളരെ സജീവമായിട്ട് നിൽക്കുന്ന സഭയുടെ പ്രസിഡന്റ് ശ്രീ രഘു വർമ്മയ്ക്ക് സഭയുടെ പേരിൽ കൃതജ്ഞത അറിയിക്കുന്നു. അതുപോലെ ഇന്ന് ഏകദേശം 1.5 മണിക്കൂറോളം വളരെ നല്ല രീതിയിൽ സിന്തറ്റിക് ബയോളജിയെക്കുറിച്ച് സ്പീച്ച് നടത്തിയ, വളരെ നല്ല രീതിയിൽ പ്രസൻറ്റ് ചെയ്ത സൂര്യനാരായണൻ എന്ന ചെറു പ്രായത്തിലുള്ള 21 വയസ്സ് മാത്രം പ്രായമുള്ള മിടുമിടുക്കനായ സൂര്യനാരായണന് സഭയുടെ പേരിൽ നന്ദി അറിയിക്കുന്നു. ഇനി ഉയരങ്ങളിലേക്ക് എത്തട്ടെ എന്ന് പ്രാർത്ഥിക്കുന്നു. ഇന്നിവിടെ പങ്കെടുത്ത എല്ലാവർക്കും സഭയുടെ പേരിൽ കൃതജ്ഞത അറിയിക്കുന്നു. നന്ദി, നമസ്കാരം.
പങ്കെടുത്ത എല്ലാവർക്കും നന്ദി. സൂര്യനാരായണന് പ്രത്യേകിച്ച് നന്ദി.
One more doubt.
എളുപ്പം വേണം. ഡൗട്ട് വൺ മോർ ഡൗട്ട്. ഓക്കെ.
ആരോ എഴുതിയിട്ടുണ്ടായിരുന്നു, മനേക വർമ്മ.
Maneha, you can ask now. You can ask.
No need to write.
Yeah, you have alternative ways to do SynBio. You can do SynBio. So I don't know how internships works for engineering students, but once you've gotten your degree, you're pretty much free to do any work that you feel like you want to do. So obviously the first few years, no, you don't have to you don't have to come into an ICER or Iisc or NICER for this matter. Like, you're doing a biotech degree, no? Like, you can just simply finish your biotech degree and then do synthetic biology post graduation. Or you can, since you have a biotech degree, you can also work for a PhD. I don't know the logistics of it, but if you can work for a PhD in synthetic biology, you have your job right there. Like, that is, if you have a synthetic biology PhD thesis, you're pretty much set in the field of synthetic biology. So, what is distance courses? Oh, are you talking like long distance, like you don't want to attend the university physically? Oh, are you sure you need a master's in science for PhD? I thought technology people could also take PhDs. Oh, then but like still you can like make connections with people in the synthetic biology department. Like, anyone who knows about people who work with synthetic biology, just stick to them and make sure that once you've graduated, you get to work with them. Like, you can try doing internships with people who do synthetic biology. I'm pretty sure biotech is very similar. But like, I don't know though. I'm a physics major, so I don't know the exact difference between biotech and synbio. You're welcome. Also, Vivek Varma, what is distance courses? Is it like you do not want to attend the college physically? Is that what you're asking?
Yes.
Also, have you tried like checking Coursera and other sites for certified courses in SynBio? I'm pretty sure if you get a certified course from there, you can get like a diploma. Normally, yes, like you need a bit of lab experience to perform SynBio. You need lab experience to actually do it. Just theory isn't enough. So internships where you actually work in a lab would be much better than distance courses, because distance courses can't exactly give you the lab experience. And lab experience comes more so than the theory, because you can learn the theory in like 10 minutes, but when you apply it in a lab scenario, you might just keep failing for over weeks. That's what happened with my wet lab teammates. Like they couldn't figure out what was happening. Even though they knew the theory, they couldn't exactly do what was mentioned in the theory as such. You need lab experience in order to make some strides. So distance courses were like you can get it for namesake. Like I did a course in synthetic biology, but internships where you work in the lab would be much better.
This time.
അപ്പോൾ നമുക്ക് നിർത്താം. 9 മണിയായി. ഇന്ന് പങ്കെടുത്ത എല്ലാവർക്കും നന്ദി, നമസ്കാരം. സൂര്യനാരായണ നല്ലൊരു ഭാവി ഉണ്ടാവട്ടെ എന്ന് പ്രാർത്ഥിക്കുന്നു. And we are also proud of you, your achievement and your team spirit also. Together everyone achieve more. That is team.
All the best. All the best, Surya Narayan.
Thank you.
Thank you all. Good night.
Good night.
Bye.